Good afternoon. My name is Santiago Nicolás Urtuzuástegui Peña, and I am in charge of Dr. Claudia Mancilla Simbro. Well, um, I am going to talk about the midterm report for our project that is named From Ion Channels to Vision Loss, a study of inherited retinal dystrophies at the cellular level. To introduce myself, I am named Santiago, like I said in the last slide, and I am studying medicine in Chihuahua, in Mexico. And my dream is to become an ophthalmologist, and in the weekends I work as uh, watching dogs. Also, my hobbies are watching the sky, like the stars and the moon. Also, I like to uh, take pictures. So I love all about the light and refraction, all the stuff. Well, uh, before I uh, ex- uh, explain the problems that we aim to solve, um, I would like to explain what are the IRDS. They are, they mean inherited retinal dystrophies, and they are a group of diseases from different mutations that end up in irreversible blindness. And they, well, in different articles, they are at a lot of different mutations, like we're going to see in the next slide. But in this case, we aim to determine the significance of RCBT1, especially the mutation of the deletion of adenosine in the position 707 in the Mexican population. And with this in mind, we aim to investigate the potential therapies for treating, the patients with this disease caused by RCBT1 mutations. Well, what has been done to date? Like I said, there are a lot of articles, a lot of papers that uh, report different mutations that cause IRDS. In the Mexican population, Villanueva, Mendoza, and their group reported a lot of different mutations, and the most common of them were ABCA4 and USH2A. However, in this paper, RCBT1 was not reported as a mutation in this Mexican population. However, Huang and their colleagues published the effects of RCBT1 deficiency in the retinal pigment epithelium. They measure the cilium length, and they developed an adeno-associated virus gene therapy that restored the RCBT1 expression, and they compare the cilia length of the uh, pigment epithelium before this therapy and after this therapy, and they showed that the cilia was significantly more um, large than with the RCBT1 mutation. Well, our approach to solve this problem is to design specific primers to the target mutation, that is the deletion of 707A, and we uh, designed them with Snapchin software by Dotmatics. Additionally, we design a phylogenetic tree of RCTV1 to determine or to attempt to determine the origin of this mutation. And these were our results to this part of the project. We can see here in the bottom of the slide that we designed these primers, and they were specific to the mutation that we were looking for, and they also were they don't show um, well, non by well, they showed no specific findings. So they were all very specific for this mutation, and we can see here in the electrophoresis that the mutation is showed. So this is called in silico PCR. And in the left part of the slide, we can see the phylogenetic trees, and uh, like we thought Homo sapiens, RCT1 was very, very, very similar to all the primates, like gorilla, and troglodytes, and their similitudes were like 100%. So um, we can explain this, showing that RCBT1 is essential to the retinal cell um, health. And the next steps that we aim to take is to model the mutated protein using the UCSF Chimera software and compare it to the non-mutated protein. And with this comparison, we can attempt to determine its implication for visual health. And that's it.